Last to spring 2014 clinical chemistry. This is lab basics. Like we discussed, water is our favorite reagent in the clinical chemistry lab. And like I just said, water is the most important and commonly used reagents in a clinical laboratory. Sorry, I don't know if I can keep that up. So, because it's the most important, we need a good water supply. Significant errors, you guys know, will be introduced via contamination, right? So, poorly designed purification systems can actually add contamination issues. So, like for the DI systems, that needs to be constantly circulating. Do you know why? If you have stagnant water, what happens? Well, it can pull ions from your pipes, but stagnant water is more associated with biofilms. Huh? Legionella? Yeah, Legionella. So, you know, I heard a story about Legionellas once. It was an outbreak at a VA hospital.
Yeah, that's where they named it.
That's where they named it, and it was coming from the fountain, right?
It was coming from the AC.
Well, there's the fountain was contaminated with Legionella, so it would make it airborne. The spray from the fountain.
That's why HVAC has completely been redesigned over the last 25 years.
Cool.
It's because of that.
Water tower.
Water towers?
Air conditioning systems.
Yeah. But anyways, purification systems have to be designed, and they got to be designed specifically for the facility that it's operating in. Do you know why? Where's the water come from for it? It's the feed water is going to be different in different locales, right? So, but here's some examples of some contaminations. I used to work with this guy. He works up in Gainesville, Papke. He kind of looks like Jerry Garcia. Seriously, he drives a motorcycle, but he's a hard ass. But he studies Alzheimer's disease, specifically alpha-7 nicotinic receptors, and he was studying those, and he kept getting these binding events in his negative controls, and then found out that the plastics in the centrifuge tubes that they were using were leaching this compound. So he got a patent out of discovering that compound because it was a really good binder for alpha-7 nicotinic receptors. So that's one example of contamination affecting things. Porous surface specifically can harbor bacteria, so you got to worry about what pipes are made of and whatnot with those. And then copper can leach ions also. And this guy is a famous Alzheimer's researcher. You know, notice I'm connected with Alzheimer's, so if I forget things, you'll understand why. But this guy, he's passed away now, but he was a pathologist, and he saw when he was doing histo of people with heart disease who died of heart disease that they would have the same pathology as Alzheimer's. So he started doing research and feeding rabbits high-fat diets, and they would develop Alzheimer's pathology also. So he got all these grants, started clinical trials with statins to treat Alzheimer's, or prevent Alzheimer's rather, and got a new prestigious position at a new university, so he moved his lab there, and all of a sudden his model stopped working. And the reason why, in the animal facility there, they used purely, pure water for the water system, whereas the old lab used, normal tap water. So they discovered it was the minute amounts of copper in the water plus the high-fat diet is what gave those animals the Alzheimer's pathology. So it kind of freaked him out. He had clinical trials going, and all of a sudden his model stopped working. So, and that was due to the water. So, like we were saying, water in different facilities is going to have to be treated different, and that's because that water is known as the feed water, right? So, quality of the feed water is going to dictate the purification process, and of course, like I said, it varies. And there's seasonal variations. We have rainy seasons, it's going to leach more, calcium and whatnot into the water supply. or dry seasons, vice versa. So rainfall, ground drainage, industrial wastes, there's seasons for that too. And geographical changes also. So, and that geographical changes are going to alter, your mineral content. The feed water we have for this facility is terrible. It comes straight from the well for the water supply for Alachua, and it's really, really hard. I don't know did you see when we turned on that hot water for the first time?
White.
Yeah, it came out white, and it's terribly bad. The sink in the little fish bowl, that water always clogs up, or the screen on the water always clogs up because of the minerals depositing in that screen. So purification processes, there's a number of different ones, and they're all good for different things. None of them purify the water completely. We have distillation, deionization, reverse osmosis, ultrafiltration, UV oxidation and sterilization, and then ozone treatment of water also. So this is your basic distillation setup. So you're going to have feed water going in. Basically, it's going to be boiled, right? We've done distillation in organic chemistry. It boils, and then what the heck is that? The steam condensates and then it'll come back. And actually, as it comes through, you have hot steam coming through and a heat exchanger, and then you have pure distilled water coming out. Although it's not exactly pure, can anybody think of anything that distillation's not going to get rid of in this manner? We're not talking fractional distillation, right? This is just normal distillation. What kind of things do you think it won't get rid of? I mean, you're boiling it and collecting the gas. What?
Organics?
Yeah, volatile organics aren't going to come off, right? So, what else is it going to remove? Or not remove, or remove. So it's going to be good for,
Bacteria?
what, can you see this? Or did I post it, I guess? Bacteria, pyrogens. Anybody know what pyrogens are? They didn't teach you that in immunology? Huh? No.
Yeah, it causes
Yeah, they started that.
Yeah. Yeah, you see it causes fever. Pyro generates fire, so things that generate fever. Particulates are good to be removed. Dissolved ionized solids. Most organic contaminants you can remove, but it's not good for removing ions, dissolved ions rather, such as ammonia, carbon dioxide, chlorine, and low boiling point organics. So volatile organics, it's not going to remove very well. What do you think would be good for removing these? So when these are in solution, they're typically ions, right? Yes. Deionization, and with these, typically before you run it through a deionization column, you have a charcoal column that you run it through, and that'll pick up your low boiling point organics. So it goes to the next slide. We're talking deionization. Does anybody know how this works? There's a picture of it right there. So what can you gather from that picture? You got sodium ions and chloride ions, and these are supposed to represent resin particles. So basically your deionization columns are basically just ion exchange columns that have both cation exchange resin and anion exchange resin. Okay? So the cations exchange for hydrogen ions, and then the anions exchange for hydroxide ions, so you just get pure water that comes off. So that's how those work. But you'll run out of these resins eventually, so you got to constantly replenish them, right? So and typically it looks like this. We can go down, go look at the big DI tanks that we have, and you have a big resin bed and the water just goes through and then forced through the resin bed and then out, and you should have DI water coming out. And deionization is excellent for the removal of ions derived from solids and gases. And it's ineffective at pretty much everything else, right? Because you have to be an ion to be able to exchange for that. However, it's often used in conjunction with carbon absorption, and that's going to help you get rid of any organics. So. Reverse osmosis. We've all heard of this. What is this? What is osmosis?
Moving of water.
Yeah, it moves across what? A semi-permeable membrane that, and how does it move?
Now it's concentration level. Now
Well,
with
no, it would go down because I guess, so if you have, but you kind of wording it kind of funny because it's a concentration of water is what you're referring to. So you have, right, got a semi-permeable membrane, and in one side you have lots of particulates or salts, lots of things dissolved. The water's going to try to flow this way until they're equal concentrations, right? So I guess the water here, the way you worded it, is less concentrated than the water here. So it is going down its concentration gradient. But it's going down its concentration gradient, trying to equalize the concentration of all these particulates. So reverse osmosis is when you use force, and you force this water across that membrane. So you trap the particulates on one side, and you get the water going on the other. But do you think this is how those reverse osmosis filters are constructed? No, because how would that work? It'd be a big pain. You'd have to have pistons that constantly push it, and then a valve that prevents it from sucking it. You get more and more water, you pump it, kind of like a piston. That's not how it works at all. And this is just showing you a picture of osmosis. How they're really constructed is this way. So they have a high velocity cross flow. You have a membrane that it goes across the membrane, kind of forces the water through the pores. And the pores don't get plugged up by all the particulates that way. So you got the permeate that comes out the center, the concentrate runs out the outside, and you have a feed that runs in, concentrates it. Does that make sense? So you get this, the water that comes right through and kind of forces it through. So that way you don't have this pumping motion you have to do through what everybody draws as your, osmosis. It's like just single flow and constant flow osmosis, reverse osmotic filter. So it's pretty much effective at removing dissolved organic, ionic, and suspended impurities, but it's not good at removing dissolved gases. So again, you'll dissolve gases, you want charcoal filter and not ion exchange. I always try to say that, but what am I looking? Deionization. So then there's ultrafiltration. A lot of times, have you guys done this in the protein class, where you filter media to sterilize it? That's basically one of the things it's used for, what I've used it for. So water is passed through a semi-permeable membrane with a defined pore size of less than 0.2 millimeters, 200 microns, and remove particulates, emulsified solids, bacteria, and pyrogens. It does not remove ionized solids, gases, and organic contaminants again. So it's basically, it's good for removing particulates or bacteria and things that are contaminated. UV oxidation and sterilization. So it can remove trace quantities of organic contaminants and bacteria. You're going to use 185 nanometers. And what that's going to do is going to convert oxidants into radicals, and it's going to oxidize with your organic contaminants, produce water and carbon dioxide. And then 254 nanometers is what you use to sterilize it. And that's going to, be absorbed by DNA, right? And cause mutations and prevent bacteria and other things from growing. So. Ozone, primarily used in industrial settings. It's good for organic contaminants. Kills bacteria via rupturing the membranes. But then you have to use UV again to remove the ozone following the treatment. So you gotta blast it with UV to remove it. And that's because it's not compatible with deionization resins. This will go ahead and destroy your DI resins. So grades of water purity. The one thing you'll need to know is this clinical laboratory reagent water, and it has to have a resistivity of at least 10 megaohm centimeters at 25 degrees C. So that's the definition of clinical laboratory reagent water, CLRWs. So that test will be on, or that question will be on the test and the final. I can tell you now, huh? But that's easy to know, right? And you'll probably even see something like that on your board exam maybe. However, of course it'll absorb carbon dioxide once you have this at 10 megaohm centimeters. And that's going to cause the resistivity to drop. So even if it drops, it may still be adequate for many applications, but you want to use it right away pretty much, right? And then there's also special reagent water category. And that can include things such as electrophoresis grade water or chromatography, HPLC grade water, things like that. And it'll say on the bottles if it's a special reagent category. So. But the one I want you to know is going to be this clinical laboratory reagent water. Okay, storage and handling of water. Again, clinical laboratory reagent grade water should be used immediately because it's going to rapidly absorb carbon dioxide. And storage containers should have low gas permeability, should be small because you don't want to have a bunch left over. Should be resistant to sanitizing agents, right? And drained and refilled every few days to prevent stagnation, because we already discussed stagnation, right? You can purchase water, and purchased water has been validated for specific uses, stated on the insert. Cannot be substituted for reagent water. Sterile water is no substitute for reagent water also. It just tells you it's sterile, not that it's not contaminated. And purchased water is going to have defined ion content, bacterial contamination, and resistivity at the time of manufacture. And of course that can change from the time of manufacture. Should be purchased in quantities appropriate for usage. And it needs to be protected from the environmental contamination and container leaching. And when open, resistivity and bacterial count should be done. So you get an idea of what you got there. And an unused portion should not be returned to the original container, right? We all know that anyways with everything, right? And also should be discarded after a week. So poor quality reagent water can cause problems with it could cause difficulty with coagulation and hematological analysis. Can interfere with immunoassays, instrumentation problems. Improperly regenerated DI resins can leach contaminants. Perfume can be a problem in the lab, right? Because perfume is basically, they're organics, low boiling point organics that are vaporized, and they'll be absorbed by certain waters and can interfere with certain assays also. Bacteria, silica, they're all been reported to interfere with bilirubin and enzyme analysis. So clinical laboratory reagent water is used for all quantitative procedures, most qualitative procedures. It's used for electrophoresis, toxicology screens, and it's used to prepare buffers, standards, and controls in the lab. So, what was the S again? Special reagent water, or I forget. No. Yeah, man, I'm right. It's hard to believe. I'm never right about anything. Like I said, it was used for specific purposes. I said HPLC is one of them. Chromosome analysis. HLA testing, what's that? Oh, you guys are in hematology now, right? Yeah. What IFV treatment?
That's in vitro fertilization.
Gamete intrafallopian transfer.
I know that's for the eggs.
Yeah. So those are like special reagent waters you'll see that are specifically for these purposes. So, quality control and testing. Water has to be monitored at regular intervals, the water of your water supply. So no single test for water purity. And testing is going to ensure the water system is functioning and acceptably under routine conditions. So you got to constantly check it to make sure it's working, right? So there'll be daily checks or weekly checks. And some tests are in-house, others may be referred out to contractors. So can anybody think of things you want to test for? No? Well, of course, you're going to test the resistivity. That'll probably be checked like probably weekly or daily. You go like to the, oh, come on, where's Andy? We'll come too.
Dying somewhere.
Dying, I hope not. We'll go look at our water system. Uneven wear, signs of fatigue. Head shaft, head coupling, the rotor, and that's what the rotor connects to. Brushes and bearings, that's in the motors. Power supply, motor and lubricant. Gasket, seals, mounts, and lubricants. Calibrate the speed. And a lot of times outside contractors do all of this. And that's it for the slideshow today. So we're going to start, we'll do like variation. Eileen signed me up for that class now. Physiological chemistry and histology. So don't sweat it. Doesn't concern you guys.
Yeah.
As long as I get through the lecture. Huh?
It doesn't concern.
So one thing you want to look for is microbial monitoring. Do you guys know what these are?
Oh, force bank.
Biofilm?
That's force bank with biofilm.
Those are all biofilms.
Yeah.
So that's a lot.
See the charged PVC pipes that send water to your house.
I don't want to.
No, you don't.
Yeah, I think this is a water supply pipes for like city water.
Yeah, that's HPV.
I think this was out of a water fountain that was contaminated with Legionnaires. So, huh?
A bunch of people died.
I don't know. Legionnaires doesn't kill everybody.
No, you just get rid of it.
Just the old sick people at the hospital that catch it.
Yeah, they're already dying.
So bacteria can both metabolize components of reagents and activating them. And they'll contribute to total organic contamination too, because they're going to, as they die, they'll break down amino acids and fats and stuff, and that'll add to your organic contamination. And they're going to alter optical properties of test solutions, right? Why is that? Bacteria are big, so they scatter the light, right? So, and that's, for the yeast and whatnot, that's how, in proteins class, that's how you measure the population, is how they scatter light.
That's also a
Yeah.
scatter.
And we'll talk more about light scattering in one of the other chapters. Biofilms, that's what these are, biofilms. And they're homogeneous or heterogeneous colonies of contaminating organisms. And what they do is they adhere to the surfaces, and they're 500 times more resistant to antibacterial agents than just free bacteria. And they form these colonies, and they're supposedly really hard to get rid of. So you want to do your best to prevent biofilms. And microbial testing should be done by the lab at least weekly, and there's several methods used. Before collecting, spigot should be open for at least 1 minute. So you want free-flowing water, right? You don't want to just test what's there right at the spigot that's been stagnated. You want the free-flowing water that you're going to get like you would be using for reagents. And like it says, this should also be done for reagent prep. So, volume collected depends on the test used. Samples should be tested as soon as possible, less than an hour at room temperature, or you can throw them in the fridge and in less than 6 hours. Vortexing can ensure distribution of the organisms. So have you guys done this in micro where you like test the water and you do several different dilutions and count the colonies like that? I did that in my micro lab.
We talked about it.
Coliform, yeah, that's one way they test for coliform contamination in water supplies. So they spiked water for us with E. coli, and then we had to do several dilutions and come up with a colony count or bacterial count in the water supply. This is actually a contact plate that you can set out and contact things and see what bacteria is or what's contaminating different surfaces. So. Resistivity. So we just checked that, and what that does is it acids the ionic content of the purified water, right? So the higher the ion concentration, the lower the resistivity. And ion exchange tanks should be equipped with an inline resistivity light, and we saw those lights. They go off at less than 2 mega ohms centimeters. Systems that supply clinical laboratory reagent water must have a meter capable of reading at least 18 mega ohm centimeters, and that's monitored daily. So, and we had that. If you looked at our monitor, it was up at 17 something at the time. 17? Yeah. So the pH, generally it's not necessary. Source of pH problems have led to product pH problems. The source of pH problems can read your product pH problems. Does that make any sense? No, it doesn't. It should be lead. So pH source problems can lead to product pH problems, right? Because you use the reagent water to, you mix with reagents to run assays. So if you have a problem with your water pH, it's going to end up, your reagents are going to have a problem. Some systems will allow for an inline pH measurement, and it depends on what you need at the facility, whether you need that or not. Pyrogens. Remember pyrogens? So they generate fever, right? Pyrogens, they induce fever. It's often LPS. What's LPS? Yeah, lipopolysaccharides. Generally not routinely monitored, but they can interfere with immunoassays and can be affected by, pyrogens. So immunoassays, you're running with antibodies. They can bind to your antibodies and interfere with those assays. Testing for these may become more important in the future, especially with more and more immunoassays being developed. And these are lipopolysaccharides from E. coli or R. What's that? Huh? Silica, it's a major problem in the water supply of some geographical areas. It interferes with trace metal and electrolyte analysis. It's readily removed by distillation and reverse osmosis. Can be measured by analysis at 800 nanometers, spectrographic analysis. So I think we have a problem with silica here. Organic contaminants, generally bacteria in resin beds will increase your organic contaminants. And the method for monitoring organic contaminants are either not specific, such as permanganate. You can test with permanganate, or impractical, using HPLC. We can test the water, see what shows up after you run it through HPLC. Good system design and maintenance is the best approach to deal with organic contamination. And semi-annual sanitation helps control bacteria, and that's pretty much where you're going to get the organic contamination is from bacteria. And use of carbon absorption and UV treatment will help remove those also. Constant surveillance ensures quality. Documentation and record keeping. When you're in the lab, you have to constantly document, like we saw back there, the QC, quality control. And if there is a problem, you have to document what you did to correct it. And so to do this, there will be procedure manuals that are developed for the water purification systems, and they're going to include quality assurance plan, defining personnel responsibilities. If you don't define what's the responsibility of whoever, nobody will do it, right? Preventative maintenance procedures are going to be defined in that. Quality control checklists, like we just saw back there. And worksheets for documentations of daily, weekly, monthly, and other testing. And documentation of problems and corrective action taken. So to move on from water, we have other chemical laboratory supplies. And you've seen these, they have these labels. And it's going to tell you the purity with the types of concentrations of impurities in them. If you guys have looked at like the metals or liquids that we have, it tells you like all the different metals that are in there at the tiny percentages or parts per million. And less pure grades are not suitable for analytical work, and they can include chemically pure, practical grade, technical grade, or commercial grade. Qualitative and quantitative analysis requires ACS specified grade chemicals, including analytical grade or reagent grade. So standards and references. So what do we use standards for in the lab?
I don't know if we turn the thing back on.
Huh? Oh, yeah. I thought Andy did that. But. Yeah. So what do we use standards for? I know you guys have done standards.
Known value.
Yeah, it's a known value of some analyte that you're going to be testing. So that's the standards and references. So basically, they're known values of substances. Primary standards are supplied with certificates of analysis, and they have to be stable, non-hygroscopic, and defined composition. What's hygroscopic? Huh? It sounds like hydroscopic or hygroscopic. It absorbs water from the atmosphere. So you don't want them to do that. So, standard reference materials are available through the National Institutes of Standards and Technology, or NIST, and they're not as pure as primary standards. NIST defines the chemical and physical properties with the certification, and they're used to categorize other materials. Can be solid, liquid, gaseous, crystalline, powder, or lyophilized. Do you guys know what lyophilized means? No?
Dry, frozen.
Yes, freeze-dried is lyophilized. So your lyophilized standard, it's going to tell you to add a certain volume, probably of reagent water or CLRW water, and it'll be at a known concentration once you add that water and resuspend it in solution. You'll see a lot of our standards that we get with kits are lyophilized, or reagents are lyophilized. The enzymes and antibodies can be lyophilized, and you rehydrate them to the set concentration and then use them. And then once you rehydrate them, they're not as stable as they were when they were lyophilized. So things are lyophilized for stability purposes. And you will see once we do the quality control chapter, you can use lyophilized control pools for making sure your lab's functioning properly. Organic solvents. So their classification follows the same guidelines with the other chemicals. Often grades are more pure than a reagent grade, and they can include spectral grade, nanograde, or HPLC grade. So these special laboratory water would fall into one of these, although we're talking about other organic solvents. Purity ensures minimal spectral interference and minimal residual contamination. Solvents are more than 99% pure with no single impurity greater than 0.2%. So it's important for spectral interference, especially when you're doing like HPLC, right? Because you'll learn when we're looking at compounds coming off an HPLC column, we typically use absorbents is the most common method that's used. Some sort of absorbents at some wavelength. So chemical safety, many chemicals and solvents are flammable, teratogenic, what's that?
Cancer.
Now carcinogenic causes cancer. What's teratogenic? Vitamin A is teratogenic. There's that skin cream they tell you not to use if you're a child, if you could get pregnant. You know what? I just gave it away. Pregnant, teratogenic. It causes birth defects. So teratogenic causes birth defects. Care has to be taken when you're working with chemicals. Inhalation of fumes or dust has to be avoided. Gas cylinders also require specific regulations. Who's seen that Mythbusters where they chop the ends off the gas canisters?
It turns them into rockets.
Yes. They fly them through concrete walls. So that's safety issue with gas cylinders. They have to be bolted down somewhere because they topple over and don't have their cap on. Just have the regulator or just nothing on them but the valve. That valve gets knocked off, it turns it into a huge rocket that can kill you. So,
Yeah, they're no joke. I've seen a welding tank fall on once ago too, and that was not.
scary.
Yeah.
I've only seen videos of, like when I was at Pitt, they made us watch, the chemical hygiene, they have little videos and EH&S that we have to watch.
Body shop, we had a big welder, and one of the guys in the video, he knocked it over, and it didn't blow up, thank God. It would have been a bad day.
Yeah, because you got acetylene there too, probably.
Yeah.
So desiccants. So desiccants are what you do if you have something that's hygroscopic. You store it with desiccants because they're used to absorb and remove water from the air or other substances. So they'll grab the water from the air rather than the substance you don't want to grab from the air. So they lose efficiency, however, once they absorb a lot of the water, and some can be regenerated via heat where you just cook the water off and then reuse it again. So. And then we use a lot of plastics and glassware in the lab. We have tubing, latex. It's good for glass connections. It's affected by oils, alkaline, corrosives, and hot water. Neoprene should not be used with chlorinated or aromatic hydrocarbons. So that'll react with the neoprene. Tigons, resistant and inert to chemicals, can be heat welded, gets discolored and brittle over time, however. And then Teflon's the most expensive, and it's really resistant to a lot of things. Types of glass include borosilicate, which is Pyrex or Chymex, low alkaline earth content. It's free of heavy metals. Liquids can be heated to 600 degrees with minimal contamination. So it's really good to work with stuff in glass. Plastics, most common plastics are polyethylenes, polyethylene, polypropylene, polystyrene, polycarbonate, Teflon, and PVC. So polyethylenes are relatively chemically inert and resistant, but can be attacked by concentrated sulfuric acid and organic acids, and hydrocarbons can penetrate the plastic. Polycarbonate's stronger and has better temperature tolerances. It's chemical resistant, but not as good as the polyethylenes. PVC is soft, flexible, and porous, so you can get things growing in porous materials like PVC. You guys have seen these before, right? Or any of these you don't know? So we've got beakers, funnels, desiccators, graduated cylinders. These are graduated cylinder burette funnels, flasks up there, volumetric flasks, that would be, this is a volumetric flask, right? Syringes and pipettes. So, who's used one of these pipettes before? So not everybody. Basically, they're real easy. At least you can use them with those bulbs, which is kind of a pain, but we have the electric ones are a lot easier. And basically, they're certified to a particular accuracy, and you're going to read it at the bottom of this meniscus, right? The volume. And you kind of suck it up how much you want, and you can spit out the exact quantities too. It's not super accurate, but it can be. And these are examples of different to-deliver pipette tips for those. And these are some of the methods, so proper pipetting technique. And this is a figure from your book, and this is one of those bulbs I was telling you about. If you guys ever use these bulbs? You have? Yeah. We got electric ones to use here. But I got a whole bunch of those too if you want to try those. Probably most labs you work in will have electric ones. And these pipettes we've all used, right? So you guys know proper pipetting techniques. And has anybody ever done quality control for micropipettes? Yeah. Yeah, you measure the weight of them at the different volumes and make sure you're within 10% or so of the volumes. Otherwise, you got to take them apart. Or typically in a lab, you'll hire somebody to do it. But you should have, if you do it yourself, you can do it yourself, but you got to have the balance. They're super expensive, really fine balances that they use. Yeah. So you either send them out, or a lot of times people will come here and do it. And these are just the steps to go through with those. And SI units in the clinical lab, we all know what a mole is, right? No?
Yeah.
So you remember Avogadro's number? A mole of a substance is the number of particles in a mole is Avogadro's number, right? So the SI unit for enzyme activity is the catal, the amount of enzyme that will catalyze transformation of 1 mole of substrate per second in an assay system. It's not been approved by CGMP. International units will continue. And you can convert from 1 catal to 16.67 international units. And when we do our enzyme assay at the end of the semester, we'll calculate the IUs per ml of alkaline phosphatase activity in your plasma or serum. Anybody know the differences between plasma and serum? You guys are just taking hematology now, right? But you've had it, right?
Yeah.
So what's the difference?
Well, plasma's with the red blood cells, and the serum is just the
Nope.
white blood cells?
Nope. All the cells are gone from both plasma and serum.
Serum
Isn't
has
plasma
IgG,
whole blood?
antibodies.
Whole blood is whole blood.
Yeah, plasma has albumin.
They all have albumin. There's one protein different between the two.
Isn't it the fibrinogen?
Fibrinogen is different. That's because with serum, you let the blood clot and you spin down the clot, and then what you have on top is the serum. Plasma, you have some kind of anticoagulant in the tube that you collect it in, and you take it, spin it down immediately, and it still has fibrinogen in the plasma. So, yeah, heparin, EDTA,
Yeah.
a lot of things. So you'll be doing that in hematology, I'm sure.
Immunoheme.
Oh, is that immunoheme?
That's immunoheme.
Okay.
Because in hematology, you don't really, you just more.
It's the blood, physiology of the blood. Okay. So balances, we have microbalances, top loaders, analytical balances, and floor scales. You guys know the differences between those, I'm sure, right? This is a figure from your book that shows you how the electrical ones kind of work, I think. So. Validation, so we have this really exciting lab we're going to do where we run through the quality control with our balances with the specified weights. It's real exciting. I love it. I know you guys will too. So basically anything you have to do, the balances have to be validated when you're in a GXP laboratory. What is GXP, do you guys know? Don't know? What is GLP? Or GMP? So GXP stands for GLP, GMP, and GCP, all at the same time. You should know, you work in one. So GMP is good manufacturing practices.
Oh, yeah.
GLP is good laboratory practices, and GCP is good clinical practices. So that's all part of quality assurance. They have all these GXP laboratory procedures you have to follow, which is documenting everything, right? So, and that's what that's about, is documenting and validating equipment. So how's it done? Monthly checks are required, minimum and maximum weight checks for the balances. The monthly uncertainty is checked using the minimum weights. Additional maintenance is done when required, and semi-annual calibration checks involve the use of approved outside vendor to perform calibrations and maintenance, and includes lubing and thoroughly cleaning the equipment. So daily use. User performs daily checks using the minimum weight accepted on the balance. This is required before the balance may be used for any GXP purpose that day. So you have to check that it's good to go before you can use it if you're in a GXP facility. Daily accuracy specification is the certified minimum weight versus the measured minimum weight, plus or minus 0.1% of the weight. Daily checks along with any other entries such as the material to be weighed or recorded in the logbook. And then the logbook is cross-referenced with the lab notebook. So types of centrifuges, swinging buckets are just what that describes. The buckets kind of swing out. We see that in the clean room we have, and the other room we have swinging bucket centrifuges. Fixed angles are just what they are. They're just big chunks of aluminum typically with holes bored in a specific angle that fit the tubes, or adapters for the tubes. So who here knows what the units are if you're going to be writing a protocol that has centrifugation in it? What? Huh?
RPM?
No.
Jeez.
Yes. And that it's relative force or relative centrifugal force to gravity. So it's the number of times gravity the force is. So that question is also on a test, and everybody always gets it wrong. Everybody puts RPM, but because that's what you see in a lot of protocols, they put the RPM, and that's good for a specific rotor on a specific centrifuge. You want to be able to cross different size rotors, so when you're writing a protocol and you're centrifuging something down, you want the G forces, so it's the same on different centrifuges. And that can be calculated by this equation, right? The relative centrifugal force is equal to the, what's this, the rotational radius, and that's going to change based on your rotors in your centrifuge, times your angular velocity, and that has to do with your RPM, divided by, the force of gravity. So it's your relative centrifugal force. So remember that. That's your what units centrifuging is done in. So centrifuge use and care, always operate with the lid closed. I don't do that. I've left. In fact, I used to have a centrifuge I had to rig so that it would always be, you could have it open or closed because the thing didn't work. Don't do that. You're not supposed to do that in the lab. You always want to balance the contents before turning on. You guys know this, right? So, and I assumed everybody did back when I was in grad school. My boss gave me an undergraduate to shadow me, and we were doing these large-scale plasmid preps there was like several liters of bacterial culture I was growing. you know, showed her the protocol and she was doing it, but I didn't show her how to use the centrifuge. I just assumed she would balance it. But she put all the tubes on one side and it ripped the rotor right off the shaft. I felt so bad for her. And I felt bad because it was my fault, right? I just shouldn't assume people know how to use things. There were no biotech courses at UF. It was just students would volunteer, and my boss would give them to me because he didn't want to deal with them. He just, here, take these people. So keep the lids on the tubes when spinning. That's not always true, right? When you use those columns, you let the lids off. And do not open the lid until the rotor has come to a complete stop. Yeah, that's all safety purposes, because you stick your hand in something that's spinning at 1,000 RPM, you don't take a finger or a hand off, right? So, huh? Yeah. So function checks for centrifuge, you want to make sure it's properly balanced, lubrication, and the rotor's functioning. And routine maintenance should include clean the interior, condenser coils, fan and screens, investigate unusual noises or vibrations. I know I used to work with these really old centrifuges, and we'd load them up, and I always have to make sure they would be okay before they'd come up to speed. I'd stand up and wait for them, because sometimes it would just be all crazy. Inspect for evidence of wear, cracks in the fittings, corrosions, uneven wear, signs of fatigue. Head shaft, head coupling, the rotor, and that's what the rotor connects to. Brushes and bearings, that's in the motors. Power supply, motor and lubricant. Gasket, seals, mounts, and lubricants. Calibrate the speed. And a lot of times outside contractors do all of this. And that's it for the slideshow today. So we're going to start, we'll do like variation